Hi, and welcome to our Disparities Talk today. First, I want to just thank the organizers, Dr. B and Norlee, for all of your hard work to get this symposium together. I'm very excited to share some of the work from the International Center for the Study of Breast Cancer Subtypes. Today, I'm specifically going to talk to you about genetic ancestry and the dark side of breast cancer disparities. I have no disclosures. This is just a brief outline. I'm going to first of all start talking about the emergence of disparities and how this unmasked tumor biology differences. Then we're going to take a global approach to disparities, and I'm going to show you how we've used race and now more importantly ancestry to uncover genetic underpinnings of differential tumor biology, and closing out with some immunology work. So as you know by now, breast cancer mortality is more than 40% higher for African American women in the US than Caucasian women. But this wasn't always the case. So over time, we actually find that historically, white American women had a higher incidence compared to Black American women of breast cancer. These lines only started converging in the last 5 years, partially because of better surveillance and earlier detection in this population. However, if we look at the mortality rates, we see that prior to the mid-'80s, it really didn't matter if you were white or Black, you had the same probability of surviving a breast cancer diagnosis. The gap starts to emerge at the implementation of endocrine targeted therapies, and where we see that the white American line starts to decline at a better rate. And this is primarily because of the fact that these endocrine targeted therapies did not benefit the the Black population as well as it did the white population. So we know that race differences in survival are due to several different types of factors, including social determinants as well as biological determinants. However, when we do that multivariate analysis, we find that if we look specifically at the biology, tumor characteristics account for almost 24% of the differences in survival rates. And so as a premise, we really focus on the fact that phenotypic tumor subtypes do lend to different prognostics, right? So outcomes. One of the categories that we look at is hormone receptor status, and the subtype called triple negative, meaning it does not express specifically those targets for endocrine targeted therapies, has the highest rate in non-Hispanic Blacks. Using genomics, we actually know that there are at least 5 different subtypes of breast cancer now. And using prognostic indicators, we can determine which of these subtypes has the best potential outcome or probability of survival. And unfortunately, triple negative has the highest graded diagnosis and the poorest prognostic, with a very limited regimen of targeted therapies available. So when we think from a global perspective, we see that, for instance, the continent of Africa, when we look at World Health Organization data, has the lowest incidence of breast cancer, but the highest mortality burden typically worldwide. And that parallels what I just described to you about African Americans and Caucasians. But interestingly, our approach to oncologic anthropology, started by Dr. Lisa Newman, who's the chief of breast oncology here at Weill Cornell, depicts a very interesting trend where West African women have a higher rate of triple negative breast cancer than East African women. And we know that West Africa is primarily where most US African Americans are descended from. And globally, when we look at triple negative breast cancer, in any country where we can separate out the African component, so African Americans in the US, Black South Africans in South Africa, we find that that component of a population has the highest frequency of triple negative breast cancer. Even within Africa, when we compare different regions, we see that the West African nations have, by and large, the highest overall incidence of triple negative breast cancer. And so what is this African connection that we see across the diaspora? could our ancestral African genes play a role in tumor biology? Well, first, to appreciate this, we must appreciate that the African diaspora is a very unique population, consider it and compare it to other human populations, which are usually defined by world migrations, where single populations simply converge to create an admixed group. In the case of the African diaspora, over millions of years and several generations, Africans were displaced across most of the Americas, as well as Central and South America. So when we look at the component of African ancestry and African Americans, we see that primarily the majority is of West African descent, but there is some additional admixture from other regions of Africa. At the ICS-BCS, where I serve as the scientific director, our goal is to assess this genetic underpinning across the entire diaspora. So everywhere you see circles across the globe indicates areas where we are actively engaged in research and acquiring research samples for our population private risk identification, as well as tumor biology differences. One of the alleles that tracks very well with the migration pattern or displacement pattern of the transatlantic slave trade is the Duffy null allele. It is a mutation in the gene called DARC. It was fixed in sub-Saharan Africa, fixed meaning over 99% of the population would have this variant of the gene because it conferred immunity to malaria in this region. And so we can see the imprint of the West African diaspora across the globe simply by looking at the distribution of this allele. In a recent publication, we actually showed that triple negative breast cancer is associated specifically with Western sub-Saharan African ancestry. On top of that, the Duffy null allele, which is at this top line all the way across, is one of the most significant risk alleles for triple negative breast cancer. And so now we utilize ancestral informative markers, which just basically mean genetic variants that are specific to subpopulations across the world, to assess whether or not ancestry of any origin is associated with different or unique tumor phenotypes. The Cancer Genome Atlas utilizes such a methodology, if you will, looking across multiple populations at different cancer sites to basically appreciate or characterize different molecular patterns that of over 11,000 patients and across 7 different data types. We now have a bigger and more comprehensive picture of the molecular biology behind tumor variation. However, the majority of the patients that were enrolled in the Cancer Genome Atlas were of European ancestry. And so we still lack the power to identify the diversity that we see in the diaspora. And so what we do at ICS-BCS is ensure that we have a targeted enrollment of a multi-ethnic cohort, and we investigate these differences in tumor biology. One of our most recent studies was published in collaboration with institutions in the Southeast United States, where we compared triple negative breast cancer gene expression patterns amongst self-identified African Americans and European Americans. But we stripped away the self-identity and instead utilized genetic ancestry estimates of European, East Asian, American, South Asian, and African ancestry to correlate with gene expression patterns. We've identified a subset of genes that have unique expression in people of substantial African ancestry. What's interesting about these specific genes is that we see canonical or well-defined pathways or genes that have been attributed to some sort of cancer risk or cancer progression or otherwise malignant biology. However, when we looked at this difference between African Americans and Caucasians, we didn't see what was the typical pathway. In other words, P53, for instance, is one of the most mutated genes in just about every cancer site that's been investigated. However, in African Americans, it appears the TP53 pathway, including its downstream targets, are activated in these tumors, which indicates that P53 has a unique variant in this subpopulation. So going a step further, in collaboration with the John Carpenter Lab at the University of Southern California, we're now looking at gene expression patterns comparing West versus East Africans. In collaboration with the New York Genome Center, we track genetic ancestry, and I'm even going to tell you a little bit about our ability to call or identify novel mutations in BRCA cancer genes. So first of all, in our African analysis, we also subdivide not only the world populations, so East, Southern Asian, European, American, and African, but also subcategories of world populations. And so we're able to not only identify the fact that, for instance, our Ghanaian populations are primarily of West African descent, but that our Ethiopian populations have a significant amount of European ancestry that typically is of Tuscan Italian descent. So this is all very interesting. But when we come back to the gene expression profiles, we see that there are a subset of genes that in our Ghanaian population show upregulation compared to our Ethiopian population and vice versa. What's even more intriguing is when we also include African Americans in this grouping, we can identify subsets of genes that correlate with the admixed ancestry in African Americans. So African Americans are a combination of African and European descent. And with this powerful analysis, we're able to detect the gene expression profiles of genes that have shared African signatures between, for instance, our Ghanaian and African Americans, as well as gene signatures that have shared signature profiles between our African Americans and the Ethiopians with significant European descent. When we look at the gene pathways, again, we see canonical pathways once more, where, again, what we would normally see as a deleterious mutation or a pathway that's been shut down. When we look at West versus East African tumors, we see that these pathways are actually activated. And so this means that the tumor biology underlying, for instance, Eastern versus Western Africans and African Americans with substantial West African ancestry could potentially have differences in the genetic networks that drive tumor biology. And now we look at African-specific alleles. So again, as part of our pathway or our workflow, one of the things we can do with our RNA-seq data is identify novel mutations across the gene. So what I'm showing you here is a model of the BRCA1 gene. And each one of these lollipops indicates a mutation that we identified currently in our Ethiopian and Ghanaian cohorts. The taller the lollipop, the more frequently we've seen this particular mutation. Now, again, we've called these mutations in tumors. And so it's important to go back to the germline to determine whether or not these are heritable mutations that could influence risk. And what we've done then is to look at the 1,000 genomes data sets across all the world populations for each one of the alleles that we identified in our sequence. And we found, for instance, one that has a very high frequency, over 80% in the African populations in 1,000 genomes germline data. We find this particular allele in very high frequency, almost 90% of the cases in our Ghanaian cohort, and over 85% in our Ethiopian cohort. When we look now within our African American versus European populations, we also see this allele found more frequently in our African American cohort. Ultimately, what we want to do with this data is give better significance to what would now be called variables of unknown significance. And we see that, for instance, when we map these mutations onto the actual structure of the gene, we can find that there would be structural changes that could implicate different function or misfunction, if you will, of the BRCA gene. And so all of this work is very important towards finding better models for survival, better models for risk that are informative in all multi-ethnic cohorts. In our tumor biology, we also investigate the immune response in tumors. Evolutionary data shows that immune cells from people of African descent compared to European descent have very distinct responses, even to the same challenge. In this case, it was a bacterial challenge. And so how much more would those differences impact the tumor immune response? Well, coming back to this DARC duffy-null allele, we actually know that the function of DARC is normally that on red blood cells, it would control inflammatory chemokines and thereby also control the influx or infiltration of immune cells, creating the tumor microenvironment. However, people who hold this duffy-null allele do not express Duffy on red blood cells and therefore lack this function. However, they do still express it on the blood vessels, and so are able to recruit immune cells, but also mount a more prolonged inflammatory event in response to not only tumors, but other infectious diseases. And what we've shown is that when a tumor is expressing DARC, the higher the expression of DARC, the more we see tumor-associated leukocytes, or the more infiltration of specific immune cells that correlate with the expression of the gene. We also see that inflammatory chemokines are more highly expressed in DARC tumors that have high expression of DARC, and that high expression of DARC is also correlated with better overall survival, where we see longer survival in patients who have high expression of DARC compared to patients who have low expression of DARC in every subtype across both overall and relapse-free survival. So all of this information is very compelling. One of the things we are actively further characterizing is what the spatial distribution of these immune cells look like. And so for that, we use a new technology called imaging mass cytometry, where we're able to look at a single tissue slide and characterize over 50 different markers. And what we find is that in DARC tumors that have low tumors that have low expression of DARC, most of the immune cells are isolated to the stromal region and don't actually infiltrate into the solid tumor space. But in a DARC high environment, we see a lot of immune cells infiltrating into the tumor space. This means that DARC is probably a prognostic indicator of a tumor immune response, and whether or not you have high expression of DARC could be correlated to genetic ancestry. I just want to close out the topic of, talking about COVID-19 impact and whether or not we're going to see disparities exaggerated in this sort of new normal. So we've seen a lot of papers come out that talk about the clinical impact of COVID-19. The study that I'm going to share with you is called the CCC19 study. We have active investigations ongoing at NYP. These are very preliminary at the moment. However, I will share that, for instance, in this multi-institutional analysis across the US, Canada, and Spain, they did identify some very compelling associations. So for instance, living in Canada is a protective factor. So anything to the left of the line means that it's a protective variable. Anything to the right of the line means that it poses a higher risk of mortality. And so comorbidities, of course, we've seen reported multiple times are higher risk of mortality from COVID-19. But specifically with regards to cancer-specific variables, this study shows that a present status of stable or even tumors that are responding to treatment, as well as present cancer diagnosis with progressing on treatment, has a higher impact or higher risk for mortality in the context of COVID. Now that said, we also know that most of these studies have been published very urgently. The data has been collected suboptimally, as many of these systems are actively trying to fight the mortality rates. And so we have to take with a grain of salt the information that we glean from these studies, including the fact that the CCC19 is limited by an observation of only 30 days and only for people who were admitted to the ICU, and that follow-up was missing for almost 61% of the patients that they had in their study. That said, as a cancer patient, it's very important to consider that you do have increased risk of mortality simply because we know that treatment can suppress the immune system and can reduce organ function. And so it's important that you do what we all do, but just more vigilantly. And that is wash your hands, don't travel if you don't have to, wear a mask, and make sure that you protect yourself from infection. Lastly, our group is starting a new project that was recently funded where we look at the dark side of COVID-19 disparities. In this study, we are investigating whether individuals who are Duffy null versus those who are Duffy positive would have a unique impact of COVID-19 because of the fact that Duffy null does not clear out the cytokines at the same rate as people who are Duffy positive. And thereby we would see increased levels of inflammatory chemokines and more tissue damage in people who are Duffy negative. And this Duffy negative population represents over 68% of African Americans in the US. Our preliminary data actually supports this hypothesis, showing that Black patients with COVID-19 positive test results have higher inflammatory markers, and I'm showing D-dimer here as an example. So higher D-dimer levels in Black COVID positive patients compared to white COVID positive patients, which we don't necessarily see in COVID negative patients. So in summary, disparities in cancer mortality is likely also driven by biological distinctions in as much as it is also driven by systemic social determinant factors. Inclusion of genetically diverse populations is key in order to unlock this diversity and characterize it. Tumor immune responses could be driven by specific race group differences, which could impact immunotherapy potentials. And our dark tumor phenotypes indicate this immune response difference as we move further to utilize precision medicine tools such as imaging mass cytometry to better phenotype these categories in our populations. And so with that, I close and I look forward to a lively, not necessarily debate, but a lively discussion. Thank you so much. Take care.